so let's get started with a sound check. Can you hear me in the back row? Hear me in the back row? Excellent. I hope that Word document didn't get lost. Oh, we don't want that one. How about this one? All right. I'd like to take your attention for a moment with some course matters. Some of them may be of more interest than others to you, but I want you to know about them so you have a choice. Angie will be leading a study group again Sunday evening at Hardin, 6 to 8 p.m. April 1st, we can't help the date. She'll tell you true things. I don't care what the date is. Susan will have a review session here on Monday, the 2nd, and that also is 6 to 8 p.m., so that's kind of easy to keep the times. And then there's the bad news. The College of Medicine is relocating our class on Wednesday. So if you come here on Wednesday at the usual time, you will not get me, you will not get an overview of metabolism. What you'll get instead is Richard Hines from MIT, who happens to be a very good speaker. So if you make the mistake of coming here instead of going where we're supposed to, you will still have a good talk. But you won't get anything towards the class. It's interesting stuff he has, but it's nothing to do with what we're doing. Just for that one day. That's right. That is the only talk. You should feel confident. This is the only speech in the whole of Research Week that they think they need this auditorium for. So go figure. Anyhow, this Medical Alumni Auditorium is way over there. And probably the easiest thing for you to do is to get on to the university's website and the maps and ask it. If I were to try to tell you how to get there, I'd say go to Boyd Tower and up to the 2nd floor and then head east. But I'll try to get a better map by Monday or before we try to do that. It's not a bad room, it's just you need to allow some time to get there and find it. And then, of course, just as a reminder, the 3rd hour exam will be a week from Friday. Do people have any questions about course matters or general organizational kind issues? Yes? I will try to have it recorded for the web. The question was, if we're medical alumni, will there be a video of it? I don't know for sure. I will certainly try to have a video of it, but I can't guarantee it because the special magical black box is on this computer, and I'll have to try to coordinate with with the MedAV. Sometimes a difficult process, but I will try. Let me write a note so I don't forget it. I know that last year when they moved us, we did not succeed at it, but we were using a different technology, and it should be possible to do it. Med Alumni Auditorium, April 4th. Okay. All right, any other general organizational type questions, problems, suggestions, resources to share? Okay. I will also circulate this amongst folks as probably an email attachment or something so you have a permanent copy. Okay. I'll wait till later, given how slow this thing is. Okay. So, lipid metabolism. Do people have any questions about lipid metabolism up through the part that we had already covered? We discussed fatty acid degradation, also called data oxidation, because the carbon data next to the carbonyl is the one that gets oxidized, or 2 carbons down from it. We talked about fatty acid synthesis, which has some similarities, but has also some contrasts. And the computer is again giving me trouble. There we go. Okay. I'm going to go straight to that is. Where? Come on. So we're not as far off maybe as we might be. 23. So we had gotten up to slide 18, is that correct, folks? Through 24? Oh, so we've covered ketone bodies? I don't think so. Right, okay. Let's see where we are. And then... Okay. Okay, so the numbering on mine seems to be different from the number, okay? So we had done through this. The numbering on my copy is different from this. Okay, there we go. There we go. This is where we are, right? Okay. I see how I got confused. Or not. There we go. Yeah. Okay. So what we've talked about so far is fatty acid metabolism, not lipid metabolism in the broadest term. Just the metabolism of straight chains of carbons with hydrogens hanging off in various degrees of saturation. There are some other branches of lipid metabolism that are also very important. Some of them are important all the time, but some of them are only important in crises or part of the time. And when we talk about what we're about to talk about now, called ketone bodies, is something that is not part of your metabolism at every moment, but it is an important part of your metabolism when it's active. Ketone bodies are a way for the body to take fatty acids and make them water soluble. You can't really carry a large amount of fatty acids in the bloodstream because there's just a limited carrying capacity. And so if you are in a situation where you need to mobilize the fatty acids as your principal source of energy, the body has to have a way to do that without deterging itself to death with all the free fatty acids. And that's what the point of ketone bodies is. It's a way to transport lipid in a water-soluble form. It also is a way of transporting lipid without using up amino acids. We talked about gluconeogenesis. Gluconeogenesis is a way for the body to make sugar when you're fasting, but it requires amino acids for the carbon backbones. And in ketone bodies, you don't need to use amino acids. You're going to make everything out of fatty acids. And so it looks something like this. Okay. You start with some acetyl-CoAs, and these are the ones that are being made from beta oxidation. You remember we were talking about beta oxidation produces lots of acetyl-CoAs, 8 for every palmitic acid that you oxidize. And because both of these have a high energy bond, these are both acetyl-CoAs, you can use that to create a new bond between them at the cost of course, the high energy bond you're giving up. So you can take 2 of these acetyl-CoAs, and an enzyme called thiolase can then smoosh them together to give you acetoacetyl-CoA. So it's like 2 molecules of acetic acid smooshed together. And you pay for it because you're releasing a free CoA molecule. So the energy for it came from the fact that it was acetyl-CoA. You add another little bit of carbon backbone to it from a third acetyl-CoA, and this is through an enzyme called HMG-CoA synthase. And it smooshes on actually to the same carbon that has an oxygen to begin with, and now you have this 6-carbon piece. It's a branched molecule, and it has an awful name, hydroxymethylglutaryl-CoA. You can see why it's called HMG most of the time. And its name actually makes sense if you think of the backbone of 5 carbons as glutarate, and then it's 3-hydroxy, 3-methyl glutaryl-CoA, but HMG-CoA is the easy way to talk about it. So now we have an extended backbone that's been made up entirely from the carbons from fatty acids, or if you had a lot of ketogenic amino acids. And so here you have your HMG-CoA. It's got the high energy bond there to activate it as an activated intermediate. And then HMG-CoA lyase can split off a CoA molecule and in the process leave a free molecule of acetoacetate. And if you look at this, first of all, it's a 4-carbon backbone. And secondly, it's obviously fairly water soluble. It's got, carbonyl oxygen there, it's a carboxylic acid there. So acetoacetate is a fairly water soluble molecule and can travel freely in the body. That's one of the ketone bodies that's present when they're needed in the body. It can also be further reduced. Here's hydroxybutyrate dehydrogenase that can take acetoacetate and make 3-hydroxybutyrate. So this is a 4-carbon molecule, and this is 3-hydroxy. There's also the possibility of having this molecule spontaneously break down to lose a carbon dioxide, that one, and end up with an acetone molecule. That's not so useful. Acetone is not useful to us. This whole process is going on in the liver. The liver is the major metabolic organ for a lot of things, and it helps address the global needs of the body. It's the one that's going to respond to starvation. So the liver can produce ketone bodies, but it's not going to use them itself. Those are then freely water soluble. They'll leave the cell, and one of the major places that it will be used would be the brain. And so the brain has to make a shift of metabolism to go over from sugar to ketone bodies with longer fasting and so on. Ketone bodies are also a sign of dysregulation of metabolism. In some diabetics, their bodies are so bad at taking up glucose into cells that the cells think they're starving, and they make ketone bodies. So usually speaking, ketone bodies are not something you want to see in your bloodstream in any large quantity. They're not per se hurtful, that they themselves don't hurt you. And there is a very interesting finding that epileptic fits, in some cases, that cannot be treated with other medications, can be controlled by keeping a child or an adult on a diet that maintains their body in this ketogenic state, where they have ketone bodies as one of their major fuels. Why that calms down the neurons, we don't know. But acetoacetate by itself is not any use. It's, freely soluble molecule floating around. What does the brain do once the acetoacetate gets there, or the hydroxybutyrate gets there? So here's a 3-hydroxybutyrate, and 3-hydroxybutyrate dehydrogenase can now oxidize it. You now have a free NADH that you can use for energy, so there's one. And we're back with an acetoacetate here. So we've regenerated the acetoacetate. And then a ketoacyl-CoA reductase can come in, and it's going to donate the CoA part from a succinyl-CoA. So succinyl-CoA has that high energy bond there, and so it's going to donate that activated CoA, so to speak. So now you have acetoacetyl-CoA again. You've got a CoA handle on it again. And it releases succinate, which stays in the citric acid cycle and gets processed in the usual way. So you're sacrificing a high energy bond. Usually you get a GTP at this point, but you don't, because you're donating the energy of that CoA moiety to the new growing acetoacetyl-CoA. And then thiolase just reverses what it did back in the liver, and now you have 2 molecules of acetyl-CoA. Okay? Is that clear? And obviously once you have acetyl-CoA, you can put that into the citric acid cycle. You can burn it. You can oxidize those carbons in the citric acid cycle. The carbons come out as carbon dioxide, and you get oodles and NADH and a little FADH2 and some GTP out of them. Any other questions? All right. Now, there's another part in the body that has a lot to do with the metabolism of lipids. We've talked about the liver's role as one of the major sites. But another one is the adipocyte. Adipocyte's just a fancy way to say in Latin fat cell. And all of us have fat cells. There's a couple of varieties of them. They provide both for energy, they provide for padding. People who've lost too large a fraction of their fat cells, when they sit down, it's uncomfy. They are capable of dividing in adults, but they don't have a fixed lifetime. It's not like blood cells, which will die and go away. But it's also not like muscles, where the muscle cell is terminally differentiation. You can never make another one. They can go bigger and smaller. So here's one that's full of lipids. If a prolonged state of withdrawal were to go on, this thing would shrink down and those lipid droplets would be substantially smaller. They are one of the locations in which fatty acid synthesis is abundant, and because of that, they have a lot of the pentose phosphate pathway. To make fatty acids, you need a lot of reducing power. You remember the cycles of reduction we talked about during fatty acid synthesis when we use so much NADPH. Fat cells are also going to have things like the elongases and the desaturase, so you can get a variety, the whole zoo of fatty acids that are going to be needed. Adipocytes are specialized for storing fat. That's their principal job in life, but many cells can in fact store fat. And under some metabolic conditions, your liver cells will have little droplets of fat in them. You don't want that to happen if you can avoid it. Muscle cells can store fat and so on. So it can be a reserve in more than one cell type, but the fat cells are the ones that are specialized for it. Any questions? Yeah. Oh, that's an interesting question. She was asking whether in extremely overweight people, whether other cells are kicking in and storing fat in a significant way. To the best of my knowledge, it's not so much that. It's rather that the fat cells they have proliferated and filled up. So that if you were to, for instance, look under the skin, you'd see in all of us there's a very thin layer of fat there. And what you'd see would be a thicker layer where there are more cells and the cells are more full. So I think it's more a matter of multiplication of the cells and filling of the cells than other cells getting full of fat. I don't think, for instance, someone who's extremely overweight has fat in their muscle cells, for instance. I don't think that's true. But good question. Any other questions? One of the things that can cause fatty liver is drinking too much alcohol. So that's just to keep in mind. Not that it would apply to any of you, but your friends might want to know. Yeah. So how does this tie in with what we eat? Because obviously there is fat in the diet, and you hear a lot of different things about fat in the diet. And I am going to say some things that disagree with the book. They may also disagree with what you've been told elsewhere. I'm happy to be corrected. If you want to correct me, come up with good controlled studies, and I'll be happy to make a public correction. It's commonly known, it's commonly accepted that saturated fats will tend to raise LDL levels. So the more saturated fat there is in the diet, the higher the LDL levels tend to be. High LDL levels are correlated with atherosclerosis. They're considered the bad kind of lipoprotein. Unsaturated fats, like olive oil, like canola oil, like walnut oil, tend to lower LDLs. If you look at the biochemical or the physiological response to trans fatty acids, they seem to act like saturated fatty acids in a super way. That is, for a gram of trans fat, you see a greater elevation of LDL than you would for a gram of plain old saturated fat. So it's kind of like a super saturated fat. Dietary cholesterol, per se, has relatively little effect on serum cholesterol or serum LDL or HDL. And here's where I'm going to start being a heretic. One of the most beautiful studies of this was done in South Africa, where they were trying to separate the question of what does cholesterol do to your blood from what does a fatty diet do to your blood. And they were trying to find someone who had a high cholesterol intake but a low saturated fat intake. And some bright person came up with the idea of egg farmers. The staff on South African egg farms tended to be paid partially in eggs. And so these egg farm workers would be eating 5 to 6 eggs a day. Aside from that, they had a typical rural black South African diet with a lot of whole foods, low meat content, low saturated fat, and a lot of exercise and so on. So these people who were eating 5 to 6 eggs a day had beautiful, beautiful serum cholesterol. So dietary cholesterol by itself is not probably the problem. It tends to be associated with a high saturated fat diet or an inactive lifestyle. Another example of a place where fat per se is probably not the marker are the Eskimos, that if we look at them, they have a very high fat diet. Eskimos get something like 70 to 80% of their calories from fat when they're eating a traditional diet. Which is interesting, the Finns also have a very high fat diet. But the serum cholesterols in those two groups are very different. And one of the differences is Eskimos eat an awful lot of cold water fish. So they have a huge amount of omega-3 fatty acids compared to the Finns. And very big differences in outcomes, almost no heart disease among the Eskimos, very bad among the Finns. So diet does affect serum lipids. But it doesn't affect it in the obvious way. Eating cholesterol doesn't give you high cholesterol. Eating a lot of saturated fat without enough of the kinds of unsaturated fat can give you high cholesterol. So, are there any questions about that? Because I know that's a little bit different than you've probably been told in the past. You can enjoy your shrimp. Shrimp is full of cholesterol and absolutely no saturated fat. So you can eat shrimp till they come out your ears and will not raise your serum cholesterol. Something that would be interesting to try to do. All right. Any other questions about lipids and diets? Okay. There are still more lipids to talk about that are of importance. For instance, we talked about triacylglycerols as our fairly small polymer of lipids. This is the molecule that's going to just have 3 fatty acyl tails, the principal storage form in an adipocyte, and in any tissue that's going to store fat significantly. We're going to need a backbone. We're going to need a glycerol 3-phosphate backbone to build it on. And we get that from dihydroxyacetone phosphate. This is actually interesting. It's an NADH, even though you could think of it as a anabolic process because you're going to make triacylglycerols. We're making them in order to burn them, so maybe that's why the body uses the NADH, the catabolic form. Acyl-CoA transferase then uses the high energy of the bond there to put on first one and then a second fatty acyl tail. So now you have a molecule that has a glycerol backbone with a phosphate sort of marker on it, if you like, and 2 fatty acyl tails. This series of reactions is obviously going to be competing with the oxidation of fatty acids. So you're either going to use this acyl-CoA for oxidation, or you're going to use it for making triacylglycerides. And it'll depend on the regulation of that, and we'll talk about the regulation a little bit. Okay. And before we can add the third fatty acyl tail, we're going to take off the phosphate, and that's simply thrown away as inorganic phosphate by a phosphatase. And then that molecule, the diacylglycerol, is going to be the target for the final acyl tail being transferred on, again by acyl transferase. Diacylglycerol, this molecule here, has other significances to the body, and we will probably be running into those in a little bit. But in this format, in this context, it's being used as a building block. And there we have the triacylglycerol with its 3 tails. There we go. We also need to somehow or other make membrane lipids. There are no dietarily essential membrane lipids. We have to be able to make our own membranes. And how do we go about that? Well, one family of them can start from either ethanolamine or choline, and then we can activate that molecule by adding a phosphate onto the alcohol of it. And at that point, you've got either phosphatidylethanolamine or phosphatidylcholine, depending on what you have on your nitrogen there. And then the activated intermediate that's going to be the building basis, the foundation for what else is going to go on, we're going to use a cytidine triphosphate. And like so many other reactions we've seen before, we're going to throw away an inorganic phosphate. We're going to split off a pyrophosphate, break it into 2 inorganic phosphates. And now we have cytidine CDP, cytidine diphosphate ethanolamine, or cytidine diphosphate choline, depending on what you have. So should look familiar as an activated intermediate. And then this molecule, since we've now activated the head group, so to speak, can transfer this head group onto a diacylglycerol. So one of the things you use diacylglycerol for is to make triacylglycerols. But the other thing you use diacylglycerol for is to make membrane lipids. And so if you do that, you have now free CMP, and here would be a molecule of phosphatidylethanolamine, or phosphatidylcholine, depending on the exact what's there on the nitrogen there. So it's going to cost us a high energy bond in order to do that. Actually, it ends up costing us more than that. It'll cost us one here, and it'll cost us one here. So it actually ends up costing us, and then one there. So it's actually ending up costing us a fair bit of high energy bonds. Here's phosphatidylserine synthesis. Here's a molecule of phosphatidylethanolamine, ethanol, ethanolamine. So here's phosphatidylethanolamine. We just made it. And we just trade off. We trade the serine for the ethanolamine because energetically those two molecules are similar enough. And now you have a phosphatidylserine. Phosphonylyl-inositol is a little different here. We have phosphatidate, which was back before we threw away the phosphate group on the glycerol backbone. So glycerol backbone with two tails and a phosphate hanging off. And this is activated using cytidine triphosphate again. So now you have a CDP diacylglycerol, so it's activated. And an inositol group can then replace the CMP. Inositol, if you look at this, is a all alcohol molecule. So inositol is almost a sugar, but not quite. Very, very hydrophilic, not charged. So anything that's got that on it has a big hydrophilic group, but no charge on it. And that releases CMP again. Now you have phosphatidylinositol. There's another family of molecules that is very strange and that has all kinds of potentials for doing both good and bad things. And those are the eicosanoids. They are signals, they are biological signals. And they are used often for local interactions, sometimes for more global ones. And what happens is that the fatty acid arachidonate, which has 4 double bonds in it, can be attacked by an enzyme called cyclooxygenase, which uses 2 molecules of O2. Now right there you know it's strange because oxygen is a nasty, nasty molecule. So cyclooxygenase can take this fatty acid arachidonate and turn it into something that has a cyclic group like this. That is a peroxide there, and then another peroxide here. The peroxidase resolves it into a simple alcohol there, but it still has this cyclic group over there. You can see why this might be a short-lived molecule. And that's good. What you want is something that'll send a message and then go away. A very large variety of things that can happen with it. This is only one of quite a large number of forms, so there are a number of enzymes that can modify that further. It's involved in things like inflammation, dilation of the blood vessels, a large number of things. Cyclooxygenase is a key to producing these, so if you can inhibit cyclooxygenase, you can inhibit the outcome of it. And one of the outcomes of some, not all, of these products is fever. And it's been known for millennia that willow bark will lower fever. And it turns out that that's because of salicylic acid. That's the aspirin that you get from whatever. Ibuprofen is also a cyclooxygenase inhibitor. Acetaminophen is a cyclooxygenase inhibitor. Vioxx, Celebrex, these are all cyclooxygenase inhibitors. If you go back and you look at the structure of it, you can see that maybe there's something in there that's mimicking an intermediate. We don't know exactly. These are cyclooxygenase 2 inhibitors, so they don't inhibit all cyclooxygenases equally. They prefer to target just the cyclooxygenase 2. Okay. And then there's cholesterol synthesis. Cholesterol is so essential to us that we make cholesterol. We make quite a bit of cholesterol. And we're going to see some resemblances between cholesterol and the ketone bodies, that there are some overlaps there between all those different things. The first steps are going to resemble them quite a lot. So you can take 2 molecules of acetyl-CoA and thiolase, get acetoacetyl-CoA, and take a third molecule of acetyl-CoA and make HMG-CoA, hydroxymethyl-glutaryl-CoA, 5 carbon there. Those steps resemble ketogenesis. The enzymes are isozymes of it, but it's happening in the cytoplasm. I may not have mentioned that ketogenesis happens in the mitochondrion, whereas this is going to happen in the cytoplasm. So it's another example where we can separate pathways by putting them physically in different places. And then we start to differ. So this step is going to be the one that is unique to cholesterol synthesis and different from ketogenesis. We take the HMG-CoA, the hydroxymethyl-glutaryl-CoA, and we're going to reduce it. So we're going to invest 2 NADPHs, that's the anabolic form, with HMG-CoA reductase, and end up with mevalonate. So here is mevalonate. You can see it's been reduced. This carbon here is the one that's been reduced. And so now we have a 5-carbon, a branched 5-carbon backbone to do other things with. This step, HMG-CoA reductase, is the rate-limiting step for cholesterol synthesis, and it's also the regulated synthesis. So the activity at this step determines the overall rate at which cholesterol and other related molecules are being made. It's regulated both by how much of it is made. It's regulated by phosphorylation on the enzyme. And because it's so important to cholesterol synthesis, it is a target for the statins. And the statins are HMG-CoA reductase inhibitors that you may have heard about. You probably can't avoid ads for them on TV. That will inhibit the synthesis of cholesterol and are associated with lowering serum cholesterols in individuals who take them. It's not as clear that they actually do you any good, but it clearly does lower your serum cholesterol. Mevalonate by itself is not the end product. It's a building block. It's a 5-carbon, 6-carbon fragment that we can use to build something else with. I'm not going to walk you through all the other steps that are needed to turn mevalonate into isopentenyl pyrophosphate. But I will show you isopentenyl pyrophosphate. And so here we have a 5-carbon fragment. It's a pyrophosphate, so you can guess that it's an activated form. It's still branched, and it has a double bond in it now. So it has sort of an odd look to it. It has very promising shape. Isopentenyl pyrophosphate is not just a precursor for cholesterol. It's also going to be a precursor for ubiquinone, and we know that that's essential in the membranes of mitochondria. It's a precursor for farnesyl group, and the farnesyl group is one of the lipids that's used to anchor proteins to membranes by having a lipid tail. If you remember back in our membrane chapter, we talked about how some membrane proteins are anchored through a lipid tail. The farnesyl group is one of those lipid tails that gets used. It's also a precursor for the carotenoids. So although we don't make large quantities of carotenoids, certainly it's an important molecule for that reason. Any questions? Mm-hmm. Let me just see where we are. Yeah, okay. This is showing you some of the structures of those molecules. And what you should see emerging from all of them is they are all branched and have aromatic bonds, double bonds in them. So these are very distinctive molecules. Now, one of the funnier things that can happen is the formation of cholesterol. Here we have one of these branched and aromatic or unsaturated molecules, the squalene. Squalene is named for whales because it's an important component of the whale oil. Didn't make that one up. And squalene can fold up and, with a number of other steps, give you cholesterol. So this long branch thing can then give you these multiple ring structures with methyl groups hanging off the sides. And that itself is cholesterol. If you look at the structure of statins, so remember these are the inhibitors of HMG-CoA reductase. Here you have HMG-CoA itself, and you can see a little more clearly what its structure might be in three-dimensional space. And you can see over here, compactin having the ring structure with oxygens hanging in that general area. Again, fluvastatin. So very likely there's enough similarity at the substrate binding site for those to mimic each other. What are we going to do with the cholesterol once we have it? Well, one place is membranes. We've talked about how membranes can buffer membrane fluidity, keeping it in a narrower range. And just plain old cholesterol can do that. The cholesterol molecule itself, with no further modification, can do that. We can also make cholesterol esters. So here, for instance, is a cholesterol ester. And it's been esterified with an acetyl of some kind. And this could either be a way to store the cholesterol. You can see deposits of cholesterol sometimes in cells under certain circumstances, or to make it more usable for transport. Cholesterol is a precursor for steroids. Cholesterol, it's an alcohol. Steroids are the family of hormones that are related to cholesterol. So testosterone, estrogen, pregnenolone, progesterone, androstenone, all of these different steroid hormones are derivatives of cholesterol. Yet another reason why we have to be able to make cholesterol. It's also a precursor for bile salts. So here is an example of a bile salt. It's cholesterol, but cholesterol that's more water soluble, or at least more amphipathic, because it's got two new alcohols here and a carboxylic acid tail. And so cholate is a very good detergent. It helps to emulsify and disperse the fats in our foods. So the pancreas squirts a little bit of bile into our stomachs to mix in with the food, and it helps disperse and emulsify the fats. Some of that is reabsorbed, some of it is lost in the feces, so we need to make some cholesterol to replace that at all times. We had alluded to the idea that LDL and HDL play some kind of a role in how cholesterol is carried around the body and its general homeostasis, if you like. It looks something like this, I mean, in broad terms. That the LDL is going to be a special package that can deliver cholesterol to cells. And so the LDL, the lipoprotein particle, remember that we had a picture of what it looked like, has to bind to a receptor on a cell. That receptor can then actually bind it and be part of a process in which it's taken up into the cell, and then the cholesterol is released inside that cell. In the opposite route, when there's more cholesterol than the cell needs, there is another membrane component, a transporter or lipase, that can sequester it and then transfer it into an HDL. And then the HDL is free to take it away to the rest of the body. This biochemistry was derived partly from the fact that there are people whose LDL receptors are defective. And in these people, the serum cholesterols can be literally 2 to 5 times or more that of a normal person. Their blood can actually look slightly milky from the incredible levels of lipids in it. So if they're not taken out of the bloodstream, they end up accumulating in it. So here's the grand overview. This is the whole pattern of lipid metabolism, the integration of everything that we've been trying to talk about. And I'm trying to decide where the best place is to start from. Let's start from fatty acids. So here we have fatty acids that have a number of possible fates. Fatty acids could be treated anabolically, taken back up to triacylglycerol, and we saw that that requires high energy bonds. Fatty acids can give us the eicosanoids, the locally active hormonal type special guys. If you make a phosphatidate, we can make glycerophospholipids from it. Or we can use fatty acids as a source of energy. And one way of doing that is through the synthesis of the ketone bodies. We can make acyl-CoAs. Sorry, this is beta oxidation shown in generic form here, I'm sorry. So you have beta oxidation as sort of the major pathway. So if acyl-CoA, meaning it could be 16, it could be 14, it could be 12, it could be any length of an acyl tail, which will give you off one round of ubiquinone reduced, it'll give you a round of NADH reduced, and then each round of beta oxidation will give you an acetyl-CoA. So this is beta oxidation. Once you have the acetyl-CoA, it can then go to several places. You can take the side route through the ketone bodies, where you can make ketone bodies and then let those pass around in the body and come back to CoA. You can use the acetyl-CoA as a building block for cholesterol. Both cases you have the HMG-CoA, but this one has a different fate to it. And through investing the more reducing power in ATP, end up with isoprenoids of cholesterol. Acetyl-CoA is also the building block to go the opposite direction. So this would be fatty acid synthesis, and we're investing NADPH at each step. And if you remember, if you want to go back and just take a look at it, you'll see that the carbon backbones are doing the opposite things here versus there. Of course, the other thing we can use acetyl-CoA for is the citric acid cycle. Be fed in as a fuel into the citric acid cycle. If we do that, the carbons are lost as carbon dioxide, and we have large amounts of NADH produced for oxidative phosphorylation. So this is the overview of all of those things. If you'd like, Angie, could you help me pass around some cards? All right. If you would be kind enough, if you feel like doing it, study questions on some of the chapters that we've covered so far. We have only one more chapter we're going to be covering before the test itself. Just to refresh your memories, on the exam a week from Friday, we'll be talking about the citric acid cycle, oxidative phosphorylation, photosynthesis, lipid metabolism, nitrogen metabolism. Clearly, you can't write study questions about nitrogen metabolism, but if you'd like to write a study question or an exam question on the chapters we have discussed so far, please feel free to do that. If you'd just like us to know that you're here, you can write your name on it, or both. In the meantime, if people have questions on the material we've covered, feel free to chime in and I can answer those at the same time. Does everybody have a card that wants one? Anybody need a card? Okay. I'll point out, and if you haven't already noticed this, that there's an overview of carbohydrate metabolism that looks like this, and we're going to have an overview of nitrogen metabolism as well. So there's places where this is all kind of drawn together and a little more coherent. Yes, sure. What farnesyl is used for? Sure. Oops. Yeah. Let me get back into here. Sure. So you were asking about the farnesyl as a membrane anchor. Is that right? She was asking about the role of farnesyl as a membrane anchor for proteins. And if you remember back, to the dawn of time when dinosaurs roamed the earth, that there were three kinds of membrane proteins, some that are completely in the membrane, the integral membrane proteins, and some that are just stuck on by charge, the peripheral membrane proteins. The third group are ones that have a lipid anchor attached to them. So there's a covalent bond between a fatty acyl tail and the protein, and the way it's attached to the membrane is just through that fatty acyl tail. So the only part that's in the membrane is the tail. The rest of it's exposed on the surface. And so it would be possible, for instance, to degrade it with proteases or something like that. Usually takes a milder detergent to remove it than an integral membrane protein as well. Are there other questions about lipid metabolism as we sort of tidy up things and close them off? I'm curious, had any of you heard about the fact that cholesterol is not bad for you? Good. The word is getting out. Good. It's really the saturated fat more than the cholesterol. Was there a comment there? What? Although, yeah, I don't think they so much label cholesterol content, do they? They give total fat compass, total fat and stuff, but cholesterol per se I don't think is labeled. Yeah, yeah. All right, thank you all. Leave the cards on your way as you go. Yeah.
Question is kind of off topic, but I was wondering last night, can plants get cancer?
Very interesting question. There are cancers of plants.
Okay.
Some of them are mediated through plasmids, little small pieces of
Yeah.
RNA or DNA. But yes, plants can definitely get cancers. I don't think they ever get metastatic cancers. I don't think there are ever cases where the cells spread. But they definitely can get cancers. And the plasmid that causes the cancer can spread. So that starting with a wound right here that gets infected with a bacterium that has that plasmid, it can shed it into the circulation of the plant so they can get masses elsewhere. You can also get things that still look like cancers from things like gall wasps, where it's kind of neat. I guess it doesn't hurt anything. They plant their eggs in the plant. They inject them,
Right.
and then the eggs release growth hormones in that area. So you end up with a great big knot of stuff
Right.
that ends up being a nice little environment for wasps to live. So yes, there are definitely tumors of plants.
Yeah, I was just thinking about that because you don't really hear about it.
You don't hear much about it.
Okay. And I have another question too. Will the majority of the mechanisms be fair game for the exam?
Well, I haven't made that the top priority. I guess is the way I would put it. In other words, I would focus your time on other things first, and then put that at the bottom.
Okay. There are quite a bit, so that's what I was wondering. All right. Thank you.
Sure.
So is the ketone bodies mobilized when our body needs energy most, like such as like a crisis?
It's mobilized when you haven't got sufficient carbohydrate. Okay.
Okay.
So when you've been fasting, it's a way that the body can mobilize the energy without doing too much damage to the muscle.
Okay.
Because to do gluconeogenesis, you have to have a source of amino acids.
Yes.
And so making ketone bodies, you can slow down on how fast you
Oh, we can spare the amino acid, right?
Oh, sorry?
We can spare the amino acid.
Well, we can spare
through ketone body synthesis.
Right, correct.
Yes, okay.
Exactly. We can avoid using up so many amino acids. That initially, you go through several phases. If you've been eating lately, your energy sources are whatever's been in your meal, basically.
Yes.
You stop eating for a bit. After about 12 to 14 hours, you start doing gluconeogenesis from the stuff that's on hand. If you don't start eating at some point after that, the body switches again to ketogenesis.
Okay.
And makes ketone bodies from the body fat.
Okay.
Because if you're doing gluconeogenesis, where are those amino acids coming from if you're not eating? Well, they're coming from your muscles.
Ah, okay.
And so the body has ways to break down muscle material
Mm-hmm.
to provide amino acids to say
Mm-hmm.
for
Which is not
gluconeogenesis.
very desirable.
And, no,
Mhm.
I mean, and then you start to go after your heart. And so you start by breaking down peripheral muscles, and then after that's gone on for a bit, the body switches over to ketogenesis.
Okay.
Um, I want to. Uh-huh. Okay, sure. Hold on to it. Make sure
So is sex hormone a steroid?
Yes.
Okay.
Those are steroid hormones, but there are also others. There are things called the glucocorticoids. So, for instance, cortisone is a steroid hormone. Aldosterone is a steroid hormone, but they're involved with other things.
Okay. So when not enough glucose is available, then keto bodies is mobilized first, and then amino acid breakdown will be mobilized.
No, no. You um, I was halfway here when you started talking.
Okay.
The first thing you do is use available fuels from your food.
Yes.
The second thing you do is gluconeogenesis,
Okay.
using body amino acids. Third thing is ketogenesis.
Okay.
Okay. So,
Okay.
so the ketogenesis is the last stage. It's not the middle stage.
So how can our body obtain the amino acid from our muscle?
Through hormones.
Through hormones?
Hormones give them that message.
Okay. I mean, where is the ultimate source of the amino acid?
It's your muscles. It's
Okay.
the proteins of your muscles are
Yes.
broken down.
Okay.
And so there are hormonal signals that the body can send that say start degrading your muscle.
Okay.
And what happens is there are proteases.
So how can it take place prior to a ketone bodies, because breaking down the muscle is not very desirable?
But it's you're we're set up to do a certain amount of that all the time.
Okay.
So there's a margin, say 5 to 10% margin
Okay.
of the muscle that you're not going to notice if you lose it.
Okay.
And so the body will go for that pool first.
Okay.
But then it knows I'm starting to go deeper than that, and then it turns up the ketogenesis.
Okay.
It doesn't turn off gluconeogenesis.
Okay.
But turns it down.
Okay.
The ketogenesis is now up.
Okay.
So you can get as large a fraction as possible from fats as opposed to from protein.
Okay. Thank you very much.
Sure.
Have a great day.
Sure, you too. Sure.
Sorry for interrupting you.
No, that's fine.
You can take care of your business.
No, that's fine. Go ahead.
Okay. So where is our next lecture on Wednesday? It's not obviously here.
Yeah, on Wednesday it's in Medical Alumni Auditorium,
Okay.
and I'll be sending this around as an email. So,
Okay.
okay,
Thank you.
sure thing. God damn piece of shit. How can it be this hard to find what's on the computer?